Biological criteria. What are biological criteria? So I think I mentioned before that the Clean Water Act talks about maintaining the integrity of, the physical, chemical, and biological integrity of our waters, and that we've done a great job on the chemical, right? We just went through all those things, all those procedures we have for the chemistry. It's great, it's fantastic. Physical, we have a few things, and biological, we have almost nothing. Most states have a narrative criteria that says, thou shalt not hurt critters. It's more elegant than that, but that's basically what it says. So this is really about biological criteria. So before I talk about biological criteria, I'm going to talk about biological assessments. There's a disclaimer, which I'm sick of by now. And so this basically says that, bioassessments are tools to really understand what the health of the stream, river, lake, coastal environment that you're looking at. It's really looking at we talk about the structure and functions, basically looking at what critters are there and what they can tell us about the health of the community. And as I said before, Clean Water Act objective, maintain chemical, physical, and biological integrity of our nation's waters. So how do we do that if we don't have some sort of bioassessment or biocriteria? And the theory is basically that, we need to maintain a balanced population of organisms that are doing the things that they're supposed to do in streams, right, or in lakes or in rivers. And so this comes out of something from Frey 1977, but I think it works. So what is biological integrity? So same definition, and we're going to just sort of break it apart. So it's the capability of an aquatic ecosystem to support and maintain a balanced, integrative, adaptive community having a composition and diversity comparable to that to natural habitats of the region. So this is where the concept of reference comes in. How do you know that this stream is the way it's supposed to be if you can't compare it to a reference stream? So that's the concept there. So also we want to make sure that we have some benchmarks to know whether something is good or bad, right? So most of us that have done some biological monitoring, we look under the microscope, we count a gazillion bugs, and we produce a table that says, we had 42 of these and 22 of those, and most people can't interpret it. Most biologists can't interpret it unless they have some sort of benchmark that says this is good and represents minimal human influence. And so we want a benchmark to compare streams across the region. Same definition, and it says comparable to that of natural habitats within the region. Now, if you look at a place like California, what is a natural region? We got tons of natural regions in California, and they're different, right? The desert is different than the mountains. And so you might have to do some classification, sort of separate, what we expect up in the mountains is clearly going to be different what we expect somewhere else. And this is really just a slide that talks about, classification. It allows you to compare like to like. And there's a lot of different ways to do this type of classification. Some is just based on, ecoregions. Right? So this happens in Massachusetts because I was too lazy to get a California slide. Oh. And this part of the definition really talks about having, a good composition and diversity of critters that we think are representative. So we got this tax list with a gazillion critters on there. what is good and what is not so good. And the things that folks typically measure, whether it's in the ocean or the streams or the bottom of a lake, are things like how many critters are there, how many of these type versus those types are there. Maybe they have different feeding groups that represent things that they do. Like, in streams you have shredders, and others you have kind of grazers and scrapers. So that tells you something about their function in the stream. You have some species that can tolerate a lot of pollution, a lot of disturbance, and you have other species that, just a little bit makes them go away. So we talk about tolerant and intolerant species. If you're looking at a fish community, maybe you catch a fish, it's a fish, it's healthy, you have 10 of them, but they all have lesions on them. That's not a good thing, right? So that's another measure that we use when we do biological things. And then, there's this idea, if we want to be like reference, we don't want to have a whole bunch of stuff that came from somebody else that's really disturbing the system. So, in a lot of our rivers, we have arundo, which is taking over a lot of our streams. So we want to make sure that we have the right balance of native species, maybe a few non-natives, but not too much that hurt things. So with human disturbance, things change, right? Climate is changing. People are, spreading out into the, the what do you call it? I can't think anymore. I've been talking all day. The interface between urban and non-urban, the wildlife, yeah. And so as we do that, we begin to affect what's happening this side. And so as we begin to affect these streams with our activities, with our pollution, with things change. So these sensitive organisms that like it really nice, they go away. These tolerant organisms that can handle anything, they stick around. The diversity tends to decrease, right? You really want a population that has a lot of different things. They tend to go down. And then you just find like sort of a weedy situation where you just have a lot of the same type of not high quality critters. Like you go out into a patch of dirt, there's a bunch of weeds. It's not a whole bunch of oaks, right? It's just a bunch of weeds that we say has less value. And so all of that is sort of conceptualized in this graph. I like it. But it's basically what I just said, right? So let me walk through it. So as you go from nice to naughty, the tolerant taxa can do well, but the intolerant taxa goes down. Species abundance actually goes up a little bit, and then it crashes down. So these are the types of patterns that ecologists look at to determine whether things are okay or not by asking the species. So most folks, that I know, are working on streams and rivers, and they're looking at these aquatic macroinvertebrates, things that turn into bugs that we don't like in the air but like in the ground. These are susceptible to disturbance, and so we want to make sure that we can tell if it's happy and know when it's not. Not only do they have benefit in their own right, but they serve as a primary food source for fish, right? And so if you like fish, you gotta like bugs. So here's an example. This is from Maine. This example has been going around since I first started, so that's how old this slide is. But this is in Maine. Susan Davies did this work, and this is the distribution of bugs in a nice, what we call undisturbed or minimally disturbed stream, right? So it's pretty nice, not too much going on. So we got some stoneflies, mayflies, damselflies, beetles, caddisflies. And this is a stream, same area that has high nutrients, and you see how the critters have changed. So you have that sort of high abundance, but you've lost some things. So just kind of going back. Dragonflies, damselflies, gone. What else do we see? Stoneflies are gone, thank you. No, stoneflies are here. They're smaller, yeah. And we start getting these things called non-insects, right? You know, leeches, worms, those types of things. All right? So, darn it. And this is from, you know, just a little drain outside of a shopping, right? So, snails, scuds, there's my leeches, midges, crane flies. So this is what we're trying to do when we look at these animals, is try to tell something about what the environment is based on the representation of these critters. And it's thought to be more integrative. If you took a sample, you know, maybe the sample was okay and you walk away that this is fine. But this is more lasting than, you know, water quality, which can change rapidly. So that was bioassessment, and now we're going to move on to biocriteria. And I'm flying through this, so if you guys have questions that I can answer, I will try. So again, the minimum goal of the Clean Water Act, or the interim goal, is to protect propagation of fish, shellfish, and wildlife. And so what we're going to talk about with the biocriteria is turning bioassessment into biocriteria. And you know, there's some places where this is happening now. I know that Region 9 is working, San Diego Regional Board is working on developing biocriteria. So what we're looking for is biologically based aquatic life uses and criteria. Right? We really want to know what fishable really means. And so the management goal is the protection and restoration of aquatic life, the designated use aquatic life. How do you know the aquatic life is good? Do you measure chemicals? Yeah, you can, but it's easier to just ask the bugs how they're doing. And as I said, it usually contains, you know, a list and some description of what the organisms do. And the thing that makes, so it's this thing, it's this quantifiable thresholds. Useful in bioassessments, so you know what's good and what's bad, because you have this line, but essential if you're going to develop criteria. So here's an example of a aquatic life use that was designated by somebody that I don't know. And it basically reads like you would expect it to read. We want to support, you know, fish. We want to preserve habitats, vegetation, fish, wildlife, bugs. Okay? So many of you probably have these in your standard right now. So just like chemical criteria, we can have a narrative biocriteria, which says something like, well, it's a general statement. Or we can have a numeric biocriteria, which says, you know, everyone's got to make sure that their streams are better than this. And there's kind of an in-between. So what some folks have done is they have a narrative biocriteria, which many of them already have in their basin plans or their standards package. And then they have these tools that they use, quantifiable tools that they use to interpret their narrative criteria. So you can think of it as sort of like a three-phase approach that some folks use. So this is an example of a biological criteria. I'm guessing it's from Maine. And so they have their narrative, which is much more specific than sort of the general narratives that I've seen folks have. And they have these thresholds that they're going to use to evaluate. So I want to have, you know, a good number of taxa for my cool water and my cold water. I want to have a lot of sensitive taxa and not too much tolerant taxa, right? So That's sort of the way that they think. There's something else out there. Oh, yeah. So I'm going to get to this later, but you'll see these things have attribute 1 and 2, attribute 2 and 3. Just put that in your head, and we'll talk about it just a little later.
Terry, I have a general question here. Since we're talking about bugs for the subjective and not a pollutant, when we're looking at implementation or specifically a TMDL, I'm presuming that the TMDL will be chemical-based. And in your experience, are you familiar with a TMDL to restore biological condition that's different than a TMDL for that particular parameter?
You know, I don't really know the answer to that question. But typically what you do when you have bad bugs and you want to fix it, right? You do this thing called a stressor identification, where you ask the question, what's causing this to hurt the bugs? And then you identify the parameter, and then you write some TMDL for that. It's very much like a TIE, if folks are familiar with that in toxicity. A TIE is you get a toxicity hit, and then you do a bunch of chemical modifications to figure out what in this sample caused the toxicity. And if you get it enough times, then you start doing something called a TRE, which is a toxicity reduction evaluation, figuring out what you can do to reduce the toxicity. So the concept is very, very similar.
So it has functional value. It's not just a redundancy of developing a TMDL that you would otherwise develop for a particular pollutant.
Yes.
Okay.
So these are basically the steps in developing biological criteria. They're very similar in concept to all the other things that we've seen. You know, have standardized methods so that the data that you're using is comparable. We may need to classify using either ecoregions or some fancy statistical techniques that classify. Identify reference sites within each class. Do the bioassessments. Derive criteria. Oops, I'm sorry. This MMI is a multi-metric index. So there's different ways of developing these criteria, but that's just one of them. Select indicators that are responsive. So if you have like 1,000 indicators and a lot of them are doing the same thing, select the most responsive so you have a good signal-to-noise ratio. And then develop biocriteria for each aquatic life use. So it might not just be general aquatic life use, but it might be, cell-monitored aquatic life use. It might be, I can't think of any others. Oh, they had cool and cold water in that one. You might have a warm water one, simply because a lot of folks' standards have cold water and warm water. So you might want to have different biologic objectives for those two. And then finally, once you've developed all these, you can apply them to your water bodies. So there's lots of ways to develop these things. One way is this multi-metric approach where you kind of say, what metrics am I looking for? Let's select for them. Let's put them together in this matrix, and we'll have a single number that reflects, sensitivities, intolerance, and that kind of stuff. Another way is to do these things called predictive ecological models. And there's a couple of examples here. One is Maine, where they did a linear discriminant model. And the other one is this thing called RIV-PAX, which is used now in California under a different name. It's called the OTE. And what that really means is every place in California is different. And so you don't expect the critters to be the same everywhere. But a nice stream in the San Gabriel might be the same as a nice stream in the Klamath. And so rather than try to classify, use this model to evaluate the types of things that cause a certain type of community, and then predict how many of each critter you expect to find at a single place, and then model how much you find. So if you expect to find 100 of a certain kind and you only find 8, then that means something is wrong. So that's basically the concept. Observed to expected, yeah. And so, the one in California, it's used in Colorado, it's used in a number of places, has been, quite powerful in sort of dealing with this. Do I have to classify every stream in California or in the nation? There's also one that was done for the National Aquatic Rivers system for the entire nation. This seems pretty self-explanatory. Sample, test your criteria, figure out if it's impaired. If it is impaired, figure out what's causing the impairment, and if you have to, implement corrections and continue monitoring. So it's pretty self-explanatory. So now we have some fun pictures, probably taken in the 70s, I'm guessing, of people sampling using a SWRCB sampler, KickNet. Yeah. What year do you think this is? 83, okay. Over here we have people sampling in, I'm assuming, a lake. They're looking at phytoplankton, zooplankton, those types of things, chlorophyll. Fish communities, either electroshocking, seines, trawls. There's lots of ways to collect these things. Okay, so I told you that this was going to go fairly fast because I'm nearing the end. So remember I showed you that diagram that looks sort of like this, and I talked about those attribute 1, 2, 3, 4. So this is a generalized concept of how things change in response to stressors. This is, any random indicator, so it could be like fish, it could be bugs. And this is a generalized stressor indicator. And so things sort of change. This one looks different than the one I did before because things look different. And the idea is that how do I explain this in a way that is nice and easy? So this is a very, very generalized picture of what I just showed you. And the thought is that, as you increase exposure, these things happen on the right-hand side. And they can change in a very predictable way. And so the thought is that you can identify where you are on the stressor gradient. So if you have really, really good values in bugs and really, really low pollutants, you're kind of up the top, and sort of the vice versa. The data usually doesn't look like that. It's usually dots all over the map, but the pattern, the edge of the pattern is always pretty much the same. So that's important. Yep.
When thinking about this from the 303D perspective, what are you listing for here? Would you be listing for the stressor based on the criteria? And if so, how do you determine what that stressor is? I mean, you have to go through the you talked about the stress identification process.
Yeah. So it's all dependent on your listing methodology and the rules that are established when you develop your biocriteria. So right now in California, it says you can list for bad biology, but you have to have an associated pollutant with it, right? That's one way it could go. If we believe that these things are really accurate in determining whether something is good or bad, and the criteria is developed in such a way, then these can be use to independently place something on a 303D list. I'm not meeting the aquatic life use because I have bad bugs or I have bad fish or I have that. The next step is really the stressor identification. Why am I having a problem? Okay. So I showed you that gradient, and so some folks have divided it up into these sort of 6 generalized statements of those environments. I have to catch up with myself. I guess I don't have it in here. All right. So basically, you know, a natural system all the way to extreme changes and really bad stuff, right? And there's things in between. And you can break this down further to say what attributes are making these things be 1 up high or 6. Let me just go back. You know, what's making it natural and what's not making it natural. So folks are beginning to use these attributes to sort of identify where you are on that generalized stressor gradient. And folks are beginning to think about ways to use this to classify expectations in different water bodies. So this works best in places like Maine and Ohio, where they have sort of a different way of defining uses. So in California and a lot of our states, we say, you know, aquatic life, and these are the numbers. Some states have, these are A waters, these are B waters, these are C waters. And to A waters, we ascribe all of these uses. To B waters, we ascribe a smaller set. And to C waters, And so what Maine and Ohio do is they use this information to place waters into A, B, C, and D, and then they track over time in their 303D report how many waters are moving up this slide. But it's kind of a useful management tool to say we're making improvements in aquatic life, and waters that we used to say were pretty bad are now good. I think that's what I'm having here. And so this is what Ohio did. And so they basically have these 4 classifications, from exceptional water to limited resource waters. In between, we have a modified warm water habitat. Does this look familiar, irretrievable human modifications in physical habitat? Where have we seen this before? Anybody? Huh? In the UAA discussion, right. So this is how they are sort of doing that. Now I'm becoming Deborah and messing these things up. Yeah, go ahead.
Let's see if I can get this close enough. So it sounded like you said, at least with Maine, they're trying to move up the biological condition.
And Ohio, yeah.
Ohio as well. So there are mechanisms in place through the regulatory programs and other things that it's not that you're in the limited resource waters and you're going to stay there forever.
Yeah.
Do you have any idea what those mechanisms are? Is it
I do
TMDLs?
Not. I do not. And so those 4 classifications, they apply to these different regions based on what's happening in there. And so they have these areas that's the Corn Belt. They don't expect the same in the Corn Belt as they expect in, I don't know, Ohio, Erie. So that's sort of the concept that they use. And then for each of those areas, they have these classification that we just talked about and numbers for each of them. So that's how they do it. And Maine does something similar. So these slides, again, are ancient, but they tell a story that, if you just look at the chemical data. You see a certain story. So in this story, you know, I don't think red should be no impairment. I think it should be green. But it's basically evaluating where you get one story from one and another story from bugs, and they're different. There's places where they agree and places where they don't agree. And I hate this slide, but basically, this is what Florida is doing to communicate the results to the public. We'll have a vote afterwards. So let me get to my place here, because we're wrapping up in my half hour. Yeah, I'll just read because I can't say it. So we determine that a water body is degraded and is needing protection, so then what do we do? And this is the question that people have been asking. And it's basically the stressor identification. Again, completely analogous to a TIE and a TRE. Yeah, there's a question in the back.
That stressor identification is often inconclusive and suggestive at best, unfortunately. So that is a limitation of.
Yeah, so there's some really cool things that are happening in California to make this better, right? So the old paradigm is, I have this value in my little watershed. I'm going to look for another value in my watershed to compare with. And sometimes you had it, sometimes you didn't. But because we have this SWAMP program that monitors up and down California and has been doing it for 19 years, something like that, we have the database to now generate automatically comparing sites to sort of really tease out what is causing the impairment at this one site. So if you have all these same things and they're not causing impairment at all these other sites, you know, so I think there's good things coming. It's not quite here yet, but I think there's some future here. Yeah.
How do you decide what a reference stream is for, say, a concrete channel? Like, it's never going to be a pristine. So how does this apply to urban areas?
Yeah, that's a hard one. And we have spent years sort of talking about that. So one way is classification. We're going to say, we're not going to expect a concrete channel to be like the Klamath River, right? But we're going to expect that concrete channel to be the best damn concrete channel it can be, right? And there are variations in modified channels, right? Some are just boxes. Some have riprap with, you know, soil and dirt on the floor. You know, so really, the challenge for us is to identify what is the highest attainable use, right? And then shoot for that. And Rafi Mazur from SWRCB has a really, really nice tool that sort of helps you look at the range of possibilities for each stream and to identify where your stream is. So imagine the range of possibilities like this. This is good and this is bad. And you're here. Then this means you have possibility for improvement. On the other hand, if the range of possibility is here from, you know, really, really bad to bad, and you're here at the top of that, you can't go much further. So that type of information, I think, is useful for planning, for people just beginning to think about what can I do and how can I move the needle a little bit further. Yeah, Gary.
So ideally, biological criteria would be adopted in state standards in regulation. It'd be a water quality standard.
Yeah, that's a choice, but you know, I think it's a good choice.
But that would be the ideal.
Yeah,
I guess. A permit writer must write a water quality-based effluent limit that derives from and complies with water quality standards.
Yep.
How do permit writers write a permit limit for a biological criterion?
So, you know, these are things that need to be worked out. No, but I think it's a good question. So, effluent limits are usually about chemicals, right? And we monitor to figure out what the health of the stream is. So you might have, you might be meeting your effluent limits the entire time, but you're not meeting your biological expectation at your site. So we need a mechanism to say, there's something wrong that we're not measuring. Just like Deborah talked about toxicity, we have pollutant chemical criteria for about 126 chemicals. There are more than 126 chemicals out there. I mean, there's a gazillion chemicals out there, right? And we just don't know what they're doing. So toxicity is one way to see whether they're having an effect that's immediate. And biocriteria is another way to see whether the sum of all these things is having an effect.
But that's an assessment tool. So that's assessing your waters. Ideally, you have a permit limit to prevent an impairment. You don't want the impairment to occur.
Yeah.
So, how can, are there tools available for permit writers to be able to incorporate these biological criteria in their, when they derive the water quality-based effluent limit to prevent those impairments from occurring in the first place?
Again, that's very, very hard, and this is why it's taken a long time. So let me just take a step back. So folks have been doing bioassessment forever, right? But bioassessment only makes a difference to the people who read the bioassessment report, right? Biocriteria makes a difference to everyone trying to comply with the criteria. So that's just a general statement. So folks can do bioassessment if they want. It's great. I'm not admonishing the use of bioassessment. But if you really want to take the next step in protecting the biological integrity of our nation's water, I think you need to have some sort of biological criteria. And yes, there are bugs. We got to figure out, bugs. Yes, there are issues that we need to work through, but I think we need to be creative and stop thinking about pipe-centric effects, because it's more than just pipes that affect our environment. And again, just like TIEs, stressor identification is a way for us to identify those things, whether they're from a pipe or for a non-point source, to make the world a better place.
Terry, tagging on that question.
Sure.
Not being pipe-centric per se, but what about also not being standard-centric, where there are these inherent difficulties, and constructing the utility of this through the program of implementation to support uses as opposed to establishing objectives that lead us to a quandary of unknowns for implementation?
Yep. So, as you may have inferred, there's an issue in California where Regional Board 9 in San Diego is trying to develop biological criteria, and the issue of implementation is basically raising questions about how we do that. We don't have all the answers, but this is an intro course, so I'm just going to dodge it. Oh, yeah. Sure.
So sorry, one last question. So has EPA actually developed any biocriteria that they've promulgated?
No, we have never promulgated biological criteria. Yeah. This is to the question back there about how complicated it is. Nutrients are complicated, bugs are complicated, because again, it's not just a pipe, it's a lot of things that can affect bugs. I think I'm sort of going over my time, but you guys have the slides, you can read these slides. A lot of things affect the biological endpoint. There are some tools underway and existing that help you in the analysis. We already mentioned the stressor identification here, but there's some other tools there. There's some other resources that you can have, and that's that. I thought I could keep it to 30 minutes. Really, I did. Okay, any other questions?